Hi, so my name is Daniel Greif, and I'm going to speak to you briefly about our research. So I'm an assistant professor in the cardiovascular section in the Department of Medicine at the Yale School of Medicine. And the research I'm talking about was done in conjunction with a number of other investigators, and a lot of it was done in the laboratory of Mark Krasnow at Stanford University. The problems that we focused on are related to a couple diseases, and one of them is atherosclerosis, also known as coronary artery disease. Another disease which is related to what we work on is known as pulmonary hypertension, where the blood pressure in the pulmonary arteries which supply blood to the lung is too high, or known as hypertension. So in order for me to introduce how our research is related to those diseases, let me tell you a little bit of the structure of a blood vessel wall. The structure of a blood vessel consists of an inner layer of endothelial cells, multiple layers of smooth muscle cells, and an outer adventitial layer, which is a layer of connective tissue, loose tissue. In these diseases such as atherosclerosis, one key component of the disease is you get these smooth muscle cell layers that are found in atherosclerotic plaques and begin to impede on the vessel lumen where the blood should flow. In addition, in pulmonary hypertension, smooth muscle cells are also an important player in that disease. Usually in the vessels of the lung, you have a smooth muscle cell coverage to these blood vessels to a certain size or caliber of the blood vessels. But as you go further into the lung and the vessels get smaller, they should not normally have a smooth muscle cell coverage. Unfortunately, in this disease, they accumulate smooth muscle cells, and this impacts the disease and is an important component of the problems that people have. We basically wanted to look at the process of how you build the layers of a pulmonary artery in the mouse during the developmental period. And what we found is that there was a radial patterning. And what I mean by that is that the first layer of smooth muscle cells would form first, and it would go through some distinct steps. And while that process was going on in the first layer, the second layer would then begin to go through those processes. And so it's an iterative and successive process that eventually goes through a number of steps, and then it reaches the outer layer, which goes through some of those steps but stops before it completes all those steps. And so the inner layers that form all the steps, that go through all the steps, will form smooth muscle cell layers, and the outer layer, which went through some of those steps, will form the adventitial layer. And so through looking at those steps, we then had a platform where we could study some of the cellular and molecular processes that are important in this. And what we found is that if you can do a genetic tool, use a genetic tool known as clonal analysis, in which you can mark individual cells, we could then get an idea of where did the cells go after during the developmental process. And so we did this to mark individual cells that are in the first layer of the smooth muscle cells during this process. And we found a very interesting result, and that is that during the first few days of building this pulmonary artery, if you mark an inner layer of smooth muscle cell, it and its daughter cells, the cells that it gives rise to, will stay within this inner layer. So they will stay wrapping around the tube in this way and going down the tube in this way longitudinally, but the daughter cells will not migrate radially out. And then what we found is subsequently there's a release to this restriction of radial migration, and many of the cells will go radially outward. So the second key point that I wanted to just touch on is this radial process was suggestive that there could be a signal from the endothelial cell layer that was diffusing out and controlling the successive and radial patterning. And what we found is that the growth factor, platelet-derived growth factor B, which is expressed in endothelial cells, seems to play a key role in this radial patterning process. And in fact, we did an interesting experiment where we took beads and coated them with this PDGFB. And what we were able to do is when we put those beads into the lung, we were able to see that the initial steps of building the blood vessel wall occurred. And this occurs then in the absence of an endothelial cell layer. So ultimately, this research will hopefully allow us to become informed about what goes wrong in these diseases of atherosclerosis and pulmonary hypertension and other diseases, because we speculate that many of the processes that are important in building the blood vessel wall are important for these diseases and are also important in leading to the heterogeneity of the blood vessels that we have in our bodies. For instance, some blood vessels are much thicker than others, and some are much thinner. And so our research allows us to try to decipher what are the processes that control that.
Hi, I'm Mark von Zastrow from UCSF.
And I'm Anastasia Henry, a graduate student in Mark's lab.
Today we'd like to tell you about a study that Anastasia has carried out together with James Hislop, Joe Grove, Kurt Thorne, Mark Marsh, and me. And it concerns the regulation of Clathrin-dependent endocytosis of a particular signaling receptor by ubiquitination. Many signaling receptors, upon ligand-induced activation and initiation of downstream signaling, are regulated by a variety of mechanisms, including clustering in Clathrin-coated pits, which then mediate regulated endocytosis, removal of a receptor from the cell surface. This process of regulated endocytosis can have numerous effects, one of which is to destroy receptors by degradation in the lysosome and effectively downregulate the signal. Another is to return receptors to the cell surface by recycling, and this can sustain signaling and sometimes change the properties of the signal. So the process of Clathrin-mediated endocytosis has been studied for many years, and it's well established that it's a precisely choreographed process, regulated at many levels, from initial formation of the Clathrin coat to the subsequent endocytic scission and formation of the endocytic vesicle. The mechanisms by which this occurs involve proteins associated with the Clathrin coat itself, as well as lipids that are accumulating near the Clathrin coat. Generally speaking, endocytic cargoes, including the signaling receptors that we study in the laboratory, have thought to be passive players, essentially inert passengers in this process of endocytosis. The work that Anastasia has done has revealed a level of active control of the process of endocytosis by the signaling receptor itself, by virtue of ubiquitination of the receptor.
So what do we mean by this? We examined the question of cargo control using the mu-opioid receptor as a model, and we noticed an unexpected effect of receptor ubiquitination on the endocytic rate of receptors. This was revealed using a combination of flow cytometry and live cell imaging using turf microscopy, which allows us to selectively follow events happening at the cell surface. We found that when we prevented receptor ubiquitination from occurring, which we did by mutating the receptor's cytoplasmic lysine residues to arginines, the endocytic rate of receptors was greatly reduced. Turf microscopy revealed that when we blocked receptor ubiquitination, the receptors were still able to efficiently enter Clathrin-coated pits, but that the later stage of endocytic trafficking was affected. By following the dynamics of individual receptor-containing Clathrin-coated pits and scoring their surface lifetime, we found that when we blocked receptor ubiquitination, we significantly increased the amount of time that receptor-containing pits persisted at the cell surface before undergoing endocytic scission. We were also able to identify the specific sites that are required to regulate this process, which happen to be 2 lysine residues present in the receptor's first intracellular loop. So our findings revealed the existence of a local signaling system that is able to control the dynamics of the Clathrin-coated pit by sensing the presence of individual receptors through their ubiquitination. So after receptors become activated and phosphorylated, this leads to the recruitment of the endocytic adapter protein, arrestin. Arrestin can then link receptors to core components of the Clathrin machinery, promoting the entry of receptors into Clathrin-coated pits. Receptors themselves, we found, are able to actually stall the lifetime of the Clathrin-coated pit, effectively engaging a brake and allowing the continued enrichment of receptors to occur. Arrestin itself can then recruit the E3 ubiquitin ligase SMURF2 to activated receptors, promoting their ubiquitination. So here ubiquitin is acting as a signal to communicate information to the clathrin-coated pit to control its dynamics. This signal is sensed by the endocytic associated protein epsin through interactions with its ubiquitin interacting motifs. Epsin can then basically act as an endocytic brake release, triggering efficient endocytosis. So what are some functional consequences of this kinetic control mechanism that we've identified? Well, one function that we identified is that the lysine residues that are important for controlling the endocytosis of receptors are precisely the same sites that are required for the efficient destruction of receptors via lysosomes. So one possible function of this control mechanism could be to optimize later steps of trafficking in the endocytic pathway, basically acting as a biochemical checkpoint to stall clathrin-coated pits at the cell surface that contain receptors until these receptors have undergone appropriate ubiquitination.
A second consequence of this local signaling mechanism is that it exerts a rather strong effect on receptor loading into the clathrin-coated pit. Although receptor ubiquitination did not affect initial coat formation, nor did it affect the rate at which receptors could accumulate in clathrin-coated pits, prolonging the surface lifetime of the clathrin coat caused effectively an overfilling of the clathrin-coated pit and increased the variability in the number of receptors present in the coated pit at the time of endocytic excision. So we think another function of this kinetic mechanism is to control the surface density as a counting mechanism, essentially, to control the surface density of receptors in the endocytic membrane.
So to wrap things up, while endocytosis can clearly regulate the function of signaling receptors, our work suggests that the converse is also true, that signaling receptors themselves can precisely and locally regulate the endocytic process.